A comparative analysis of data spanning from 2020 to 2022 is also been done. We will be also documenting the microphyte growth around the lakes. Next. The following parameters are taken from KSPCB guidelines. The physical parameters we will be checking are color, odor, transparency and temperature. The chemical parameters we will be checking are dissolved oxygen, pH, electronic conductivity and total dissolved solids.
Arekere. The lake is situated at this latitude and longitude. The area of the lake is 39.53 acres. The cause of pollution for Arekere lake is inflow of anti-air sewage bottles and solid waste dumps.
This map shows here the location from where we took the sample. The first sample was taken at non-stagnant areas and the second sample was taken near the outlet point. The photos at the right side show the surroundings of the lake. Here we can see a lot of building debris and microphyte growth. Next. This table shows the results of the test we did. Electronic conductivity shows ample amount. The pH level shows slightly acidic, but it is under the ideal zone. The dissolved oxygen is almost under the is lower. The TDS also shows acceptable levels. The physical parameters, the table here shows the physical parameters. The color is always from greenish to grey throughout the study. The odor was little pungent and the temperature was 29 or 29.3. The transparency was translucent because translucent. Next.
BPM on Magnolia. The lake is situated at this latitude and longitude. The area of the lake is 272 acres. The cause of pollution of BPM lake is industrial waste discharge and sewage runoff.
Here again the map shows the location from where we took the samples and the inlet and outlet point. Again on the right side of the PPT you can see the surrounding of the lakes. Here we can again see two tables. The first one is for the chemical parameters and the second one is for physical parameters. The first one again shows acceptable levels but the pH is still acidic. It still falls under the ideal zone. Slightly basic. It's an added
The lake is situated at this latitude and longitude. The area is about 137 acres. The cause of pollution of Bearwood Lake is bathing with soap in lake, washing of clothes in lake, and dumping of building debris.
Here again there are two tables. The first one shows chemical. Here the pH level again shows basic in nature. The EC is comparatively high. Same for the TDS. It's also high compared to other lakes, but whereas the dissolved oxygen is on the lower end. The physical parameters again the same. It's darker in color. The odor is bad. The temperature was lower. The transparency was same.
The lake is situated at this latitude and longitude. The area of the lake is about 130 to 130.43 acres. The cause of pollution of Colimau Lake is solid waste dumping. Sustained inflow of anti-air sewage.
Here the map again shows the amount of the location from where we took the samples. The first sample was taken near the inlet point and the second sample was taken near the middle of the lake as the half of the lake was dried at the point when we took the sample. Again the right side shows the amount of microphyte growth and pollution in the lake.
Observation. The water quality in studied regions varies according to the location and surrounding activities. While most sites meet the basic standards for basic physical parameters, there are notable concerns regarding chemical contamination. Industrial discharges are primary sources of pollutants affecting the chemical composition of the water.
Conclusion. Physical parameters. The majority of the water samples show acceptable level of temperature with some variation in color and odor indicating localized issue. Chemical parameters. pH levels are within acceptable ranges in most samples. Though some samples show slightly acidic or alkaline nature. Dissolved oxygen show levels were average except in areas where water was stagnant. Arekere Lake. Water can be only used for irrigation and industrial cooling, controlled waste disposal or class E. There has been a slight improvements over the years. There is also a restoration project going on. Next lake, BTM and Madivada Lake. Water can be only used for irrigation, industrial cooling or controlled waste disposal, class E. There has been a slight improvement. Begur Lake. Water can be only used for irrigation, industrial cooling, controlled waste disposal or class E. There has been very slight improvement. Holimau Lake. Water can be only used for irrigation, industrial cooling and controlled waste disposal and class E. It has deteriorated. You can see here all the lakes fall under class E. Class E is the lowest in the grading. The KSPC says class A is the highest. Under the valley which we are in, which is also known as KC Valley, there are only around 3 to 4 lakes which are under class A, but whereas most of the lakes fall under class D or class E. I would like IISC for this opportunity. I would also like to thank my teachers, my coordinators and my principal for giving us this opportunity. Thank
you. You went through our literature, no? One of the thing I saw, but I think you should go through it carefully. Your pH, regarding pH, your statement is wrong. Please rectify that, okay?
Yes, sir.
And then second thing is, try to avoid reading from the paper. If you can have control over the slide, it would be better. You are struggling, especially yourself, struggling to read from the you can avoid that, okay? Try to go through the slide before and my suggestion is go through some more reports and there's a research paper. There are so many articles are there related to the thing. You put 1 or 2 slides highlighting the work done so far by other researchers. Give this one. If someone has worked on Hulimau or Madivada, you can say what was the thing when it was studied and what do they report. And why you only have few parameters, not all this one.
Sir,
you have not looked at the chemical ion.
ASBC usually does all the parameters, but there are only certain parameters from which they classify into the categories.
My question is, did you do these parameters?
Yes, we got the
Okay, then why you didn't look at the ions? Chemical ions, the anions and this one, why you have not looked at?
Sir, we did.
You have chemistry lab in your school, no?
Yeah.
Okay, you check how to do that, this one, the basic parameters. You know, ions you have to look at it and then gives an idea. See, the purpose is you have to learn. You know, what have you learned? We are not sure. By doing pH, DO and the thing is not sufficient. You need to go beyond that. Please try to do that, okay? Because anyway, it is lakes are near to your school. You can go and collect the samples and go to the lab and analyze that, okay? And also you should look at there are some comparison with the CPCB standard, BIS standard. Then based on that you should say that what category, okay? So please do that. Yeah, any questions? Yeah.
You have that project, but you have selected the different lakes where you have Arekere. Then one mistake I have got it that easy you didn't mention the unit. Okay. Then another thing that I think that now that the EC is 1,116 and the TDS is 1,005 milligram per ton. Please check it once. Okay. Then here actually your time is a lot of time you are taking in the study part. Okay. Then you are showing that study area when that result, then another study area when that result. That is not the scientific way you are doing it. Okay. You should do actually the first introduction part. Saristona, you should study some other water quality purpose and all the things. In that, what is the thing, why you want Vegu, what do you observe? Okay, then Pulimavu, that what do you observe? That is as a one table content like one or two slides, then it will be observed. Okay, then what is the water quality parameters you have selected? That one thing, it will come. Then the result and discussion part. Then what is the result you got? Then what are the discussion? That page alkaline condition, what is the reason for that? Which lake has the higher value? Then what is the reason? That everything should be come. You are in the 9th standard, no? 10th. Then you should give the better presentation. Okay? Because you wanted to compete with the other schools also. So, don't lose your mark. That's why you please prepare well and
So which lake is most polluted in our country? Which one is the most polluted? Magnolia. So in slides as in the table, you have written planting species. Okay, so in that you have written Typha, then Chlorella, Chlamydomonas, Lambda. So how do you observe all that?
We use Google. It shows on research on that.
So in which in that, what is give some example microbites? The thing is like in that slide, you have written Typha, then you have written Chlorella, Chlamydomonas. So this Typha, Chlamydomonas comes under algae. They are not under macrophyte. That's what I wanted to know whether you understand that subject or not. So please segregate that according to algae. So you write algae and then write algae leaves. Then you write macrophyte and write macrophyte. Okay? So arrange all the thing as to go because I have to spell like that. And also do the ion parameters like calcium chloride, magnesium, calcium. Okay?
See, during you were there in the orientation program, no?
I was, we were
In the Lake Symposium page, there is a student-teacher corner is there. There is a reference book is there. You know, water quality testing. In that even macrophyte details are there. How to identify that? We have given the pictures. Please go through that, and algae also we have done that. Okay? Please go through that. That becomes your effort is good. Try to improve that.
Hello there everyone, good morning, greetings to all. I am Sagar Prabhu. I am Srinath Sharmade.
and I'm Siddharth.
Today I will be discussing on the topic carbon sequestration, plants and soils. So before we start with the main topic, let's just understand how actually carbon is stored in the atmosphere and our ecosystem. This is through the process of carbon cycle. Yeah, so basically plants, by the process of photosynthesis, take out the carbon from them and from animals eat the plants and respire. Okay, so we were learning about carbon cycle. So basically plants through the process of photosynthesis exhale carbon dioxide, which is gone through into the atmosphere, and then animals also through the process of respiration exhale carbon dioxide into the soil and atmosphere. Through this, carbon is into the ecosystem, and even by burning fossil fuels, carbon dioxide will also increase in the atmosphere. And through this cycle, the CO2 levels and the global temperature and climate is maintained. Next slide. Okay, now let's dive into the main topic of the day, that is the carbon sequestration. Most of you guys must know about carbon sequestration and some might have the basic idea. So let me just summarize on what carbon sequestration is. Carbon sequestration mainly means the process of capturing and storing carbon in our ecosystem and the soil and plants. Through this, this carbon sequestration helps in mitigating global warming effects and helps in enhancing soil fertility and even the global warming and the temperature of the climate. It is done naturally through the process of photosynthesis. The plants store the carbon in their biomass, whereas by chemical and artificially the industries let out carbon emissions through which the carbon dioxide is actually stored underground. Next slide. Okay, now let's see on how carbon sequestration happens in soil. So basically, when the plants and animals die, their decomposed organic matter gets like the carbon dioxide in it gets stored in the form of humus inside the soil, through which carbon is replenished in the soil. Now let's see on how carbon sequestration happens in trees. So basically, through photosynthesis, the carbon is stored in the biomass such as trees, trunks and leaves, and through this carbon is actually replenished in plants and stored in. Next slide.
The objectives of our project are we are going to find the carbon content in soil and the carbon storage in trees. The study areas are as follows: our school, Purva Panorama and Shantiniketan Park.
So let me just tell you about the process we use to find the amount of carbon content in soil. It's called acid hydrolysis. Essentially, it's just a chemical process that breaks down a substance into its small components. We perform this method on soil to find the amount of organic carbon content in soil. Yeah, so the locations we collected our soil samples are: sample A, 10 grams of soil was collected from Shantiniketan Park. Sample B was taken from our school actually, 10 grams, right? And then sample C and D, these two samples were taken from Purva Panorama, but they are different vegetation. I'll get to that later.
Now, methodology we use to find the carbon content in trees. So basically, by each tree we used to find the height, diameter and circumference. Through this we found out the dry weight, green weight and the carbon storage from each tree species. Next slide. So the first location for the trees was from Shantiniketan Park. Now estimated of 10 different species of trees were collected. Next. So location B was from Purva Panorama, from which we estimated the 5 trees carbon content.
So I'm going to talk about the process of acid hydrolysis. So first we take the soil sample, we dry it, and then we weigh the soil sample. We have to add a strong acid to it. In our case, we took hydrochloric acid. So here we kept it for 24 hours at room temperature. Then the solution should be filtered. Next slide. The filtered solution should then be utilized. After that, we have to take the given solution. We have to heat it for some time at 60 to 70 degrees Celsius. The given residue is, we can get the given residue. So to calculate the carbon content, it is weight of residue by weight of soil into 100. So we have to, the way to find carbon content in trees are, so we have to find the diameter and height as Sagar said. We have to calculate the green weight. Here the green weight has two processes. So if the diameter is less than 28 centimeter, it should be multiplied with 0.0577 into diameter square into height. If it's more than 28 centimeter, it should be 0.0346 into diameter square into height. Now we got the green weight. To calculate the dry weight, we have to do green weight into 0.5, and then to calculate the carbon storage, we have to do dry weight into 0.5.
So these are the results that we acquired from acid hydrolysis. The 4 samples. Sample A, which is everything we collected first 10 grams. Sample A, there was 2.9, the weight was 2.9 grams after acid hydrolysis. So that's 29% carbon content. Sample B, which was taken from our school, is 21%. The 2 samples collected from Purva Panorama, the one with more vegetation, as in more plants and trees were grown on it, is 28, and the one with less vegetation is only 16. Yeah, so this is a graph here to compare. The y-axis here is the grams of soil, and this x-axis is all the soil samples. So the blue line you see is the weight before acid hydrolysis, which is 10 grams per all, whereas the red line you see is the weight after acid hydrolysis. Right? Yeah. So these are the results we got from measuring the trees in Shantiniketan Park. 10 trees from Shantiniketan Park. They are varying results. The first one was the highest one we got, the diameter of 51.3 centimeter, height of 990, and approximate carbon content that we calculated was 14,606.5 these are the 5 trees which we took from Purva. Yeah, these are the next slide. Now the analysis we see here is, so on 4 different soils, results are varying. And one of the main reasons for this can be the vegetation present on the soil. As we saw in sample C and sample D, sample C had more vegetation on the soil, thus having even more carbon content sequestrated. And of course there are other factors as well, such as temperature, time, climate around the area, and the human activity that we perform. That's right. And whereas for the trees, it's really simple. The amount of carbon storage depends on the size and shape, and the calculation freedom, like Sagar and Siddharth said, are the diameter and the height. So here the greater the diameter and height, the greater the amount of carbon stored. And that's of course an approximate value. And one more connection we found is, the more amount of vegetation on soil, the higher the rate of carbon sequestration in soil will be, and the higher amount of carbon in soil, the healthier the plants will be. Next slide. Yeah, so my friend will conclude.
In conclusion, the measures to increase natural carbon sequestration, as we have seen that carbon is actually important for plants and animals. In order to do that, we need to increase the natural aspects, and we also need to ensure that there is a decrease in the industrial emissions like carbon emissions and all. So this can be accomplished by many methods. Some of them are by planting trees like afforestation, using public transport and carpooling, educating and raising awareness to all students and all people, and using renewable energy in order to save and decrease the carbon. So these are the photos we have taken during our methods. So in conclusion, I would like to express my gratitude to TVRAMACHANDra.
Okay, we will take up the thing. See, you said number of sample you have to increase it. And how did you quantify the tree biomass?
That is there was a whole formula that we have to use.
No. Did you measure the girth and height?
Yes.
At what height the girth you measured?
Huh?
Girth, what height you measured the girth?
Oh, for that first we just found the circumference of the tree.
Circumference at what height you did?
Just
Very good, okay. Because your photograph showed at the bottom actually.
Yeah, we took it at chest level.
But your photograph is wrong then. You are sitting there, okay? And you increase the number of samples. You know, you collect the soil sample from where vegetation is there, and where vegetation is not there also you should collect. So that you can say because of vegetation how much increase has happened. That is missing. You gave comparatively, but try to do that. It's a good job. And for your information, this is a hot topic now in the globe. So if you do a good job, then you might even become the global this one, okay? So take it seriously and do that, okay?
Yes.
So,
Sir, as you said, you told with vegetation and without vegetation, right? So we are growing some radish in the soil we have taken. So we still didn't get
no, you increase the number of samples.
Yeah.
You choose this one, okay? You do that, okay? You implement it so that when you come for the lake symposium, no, your presentation would be more effective, okay?
Yes.
So I am trying to, and also that method what you did, try to have a control over that, okay? So you should understand why you are doing it and what happens. You can put that equation also, it would be good. You know, you talked about hydrolysis and thing, no? You just say that, put that equation and show that how that carbon you can determine, okay? Okay.
Sir, I have one suggestion. You have the 3 different sites now.
Yeah.
In that sites, what are the plants are there that you just give the list. Then you are taking the soil samples. In that soil sample, what is the test? Then what is the physical properties? What is the carbon?
And which type of farming?
That also you should include. Then you are taking the image, outside image now, just to give the link or reference. Okay. Like carbon sequestration that you
There was a slide with all
that slide. You are taking that image from outside now.
Okay, yes.
You are not making that it because that carbon sequestration photo that we are when you consider there maybe you are taking the soil samples.
Yeah.
So that you should mention this photo means what. Okay.
Thank you.
Jai Guru Devi, very good morning to one and all present here. Myself Anushka. My name is Julia. So our topic is observing microbial activity in the composting of organic waste and developing efficient methods for quicker composting. So throughout this presentation we'll be knowing about the benefits of composting, the importance of composting, and most importantly the activity of microorganisms in this process. So what is composting? So composting is a natural process in which waste, like organic waste, for example, food scraps and vegetable and fruit peels, these are all collected and they are added to the different layers of the soil, are covered with different layers of the soil, and we add water, fertilizers, chemicals, etc. And due to the microbial activity, the composting process is carried on. So why is composting important? Composting is important because we get to know the idea of recycle and reuse, and nothing goes waste, and also there are many benefits of composting, like it enriches the soil, and it increases the soil fertility, and besides enriching the soil, it also increases the water retention capacity.
Next slide.
The study area we chose was Swachhagraha Kalika Kendra, which is a layout plan. So here is the picture gallery. As you can see in the Kalika Kendra, there are different sections for different composting methods. So in this picture you can see home composting center. So here they have shown different machines and different items you can use for composting at home. So now we have the microbial activity of composting. Microbial activity in composting. So the activity of microorganisms in composting is very, very important, since the microorganisms break down complex substances and they convert it into water, humus, carbon dioxide and heat. So there are mainly three major conditions of microbial activity. So the first one is the mesophilic or moderate temperature. The second one is the thermophilic or high temperature. And the third one is the cooling and maturation phase. So let us know more about the mesophilic temperature. So the temperature which is above 40 degrees Celsius is known as the mesophilic or moderate temperature. So in this, the microorganisms will carry on with the process of composting. The second one is temperature which is above 40 degrees Celsius is the thermophilic or high temperature. So in this stage, the temperature, the microorganisms break down complex substances like fats, proteins, and other carbohydrates like cellulose and hemicellulose. So now let us know what happens when the temperature is above 65 degrees Celsius. So when the temperature is above 65 degrees Celsius, this kills the microorganisms, and the microorganisms cannot survive in this temperature. But we can avoid this by adding chemicals and fertilizers. So here are a few microorganisms that are responsible for composting. We have bacteria, fungi, actinomycetes, protozoa and rotifers. So here are a few methods of quicker composting. So how can we make the process of composting quicker? So first thing is keep things moving. So like we can use spades or sticks, and the compost that we have gathered, we have to keep it moving. Then adding water. So when we add water, it makes the process more quicker and efficient. And then as I said before, to avoid high temperature, we can add chemicals and fertilizers. Shred and chop. So this means that whenever we are adding the food scraps or vegetable and fruit peels, we can cut them and chop them, and then we can add them. So this will make the process even faster. And then the next one is making the environment suitable for easy activity of microorganisms. So here is a picture gallery about community composting machines. So what are community composters? Community composters are the machines
that have like the community composting machines are the machines or tanks that are very well designed aerobic tanks or containers, and these can be used in apartments and other commercial establishments and etc. So here are a few examples with the machines of used for community composting. We have the continuous composter, and then we have the shishira, and here we have the aerobic tank digester, and this is Aditi large, and that's organ stacks. So community composters are used for large scale composting. So next here we have home composting machines. So as you can see, these are very random items that we can see in our daily life. For example, we have the bucket and tubs. So these can be used for home composting.
Compost preparation activity at home. Day 1. Take a pot, put mud, sand and water. Add all types of vegetable and fruit, peels or scrap. Mix them well. And you can see here in day 30 to day 40, the microbial activity turned into compost.
So here we have the observation. So what do we observe from this entire presentation or the activity is that we learned how composting is done, and also the various composting machines. We also got to know about the different benefits of composting and how microbial activity plays a very important role in this process, and also like how to turn how to differentiate between compostable materials, like for example fruit scraps, peels, and newspapers. All these are compostable materials which can be which can turn into compost. But then plastic, leather, rubber, etc. These cannot be composted. So why we chose this topic? So the reason why we chose this topic is because we want to learn about the environment, how we want to know more about the process of recycling and reuse, and also how tiny organisms also play a very important role in balancing the environment. Here are a few references. So acknowledgement, we would like to express our gratitude and special thanks to our Director Principal Ma'am, Dr. Seema Goyal Ma'am, Jayashri Ma'am, Gayatri Ma'am, Ishwari Ma'am, Divyansh Ma'am, our teachers, and also all the other teachers who gave us this wonderful opportunity, and our parents. Thank you. Thank you for your time and attention, and we hope you found this presentation very useful, informative, and insightful. Thank you.
One other thing you need to strengthen this is there are a lot of literatures out there. You have to go through that and prepare one or two slides highlighting what is the process involved and the ages, what is the kind of efficiency they have got, all those things have to come there. Okay, now what you have done is one part of the story, but if you can bring the science to that through the literature review, that would be good. Okay, and the mechanism you have to see how that the various stages in the composting happens. You have the aerobic as well as the anaerobic condition also. So that you need to speak about it. If you can put that in a slide and then say what is your experiment and how it is.
I have 2-3 suggestions. Like first one, your introduction part, you can give the solid waste generation. How much is the solid waste generation or food waste generation, everything. Then you can have a plan. Studies also is there. That also you can include. Then in your school, how much is the food waste is there? That also you can include. Then your topic is the observing microbial activity. So you should bring the microbial. You are given the some microorganisms. In each microorganism, bacteria, what is their role? Fungi, what is their role? You should bring that one also. Okay. Then developing efficient method in your thing. Which method you are talking about that? Which one is the efficient? Then what are the method here which is there? That some more you can include. Then it will be very more wonderful project.
So here I completely felt the microbial part is completely missing in your part. So if possible, you try to identify the microorganism in that
comfort with the one by one, that you can process, it will be very wonderful.
Okay,
Try to
but
identify some, put a table on that microbe with water.
One more thing, so anyway you discussed regarding the thermophile, thermophiles now. So temperature you have looking right in the compost you are mentioning the temperature rate. So like you can also mention regarding the pH and like Sir said there are several stages in the composting. So each stages you estimate the pH as well as temperature, like physicochemical parameters. So you'll get to know like what is the condition of that compost, and in that aspect you can mention like came up with what all are the organisms that are present at that particular point. So it will be really good. So you get a good data.
okay, thank you.
Today we are going to present about the topic, role of artificial floating islands in mitigating freshwater algal blooms. My name is Nishant.
My name is Ravi.
Let us talk about the problem, algal blooms. Algal blooms refers to the rapid growth of algae, and algae is a tiny green plant-like organism that accumulates on the surface of water bodies. Excessive algal Excessive algal growth can have various negative effects, such as reducing the clarity of the water. Excessive algal growth can also decrease the amount of dissolved oxygen into the water. This will inhibit the plant growth. When the plant growth is inhibited, there is less competition for algae, and algae can dominate these waters. The decreased dissolved oxygen in the water can result in fish kills. Ultimately, all of this will result in algal bloom, and no organism can survive in these waters. Next slide. These are the current remediation strategies. There are methods like ultrasonification. It is the use of high-frequency sound waves to drown the algae. There are methods like aeration. This is to increase the oxygen levels in the water through air circulation. There are methods like mixing, which is a physical mixing of water to avoid stratification of nutrients. There are also chemical methods such as the use of algicides. There are also various biological methods such as using plant extracts and microorganisms to control the algal growth. These have their own challenges. They may be less effective, or they might be high in cost. They're not feasible to practice. They're not long-lasting, and they cannot be practiced regularly. Some methods like algicides can even prove to be hazardous to the environment. I propose strategies to use artificial floating islands, which are also known as EFIs, as an innovative means to reduce the algal blooms. Our objectives of our study is to demonstrate that artificial floating islands can control the algal growth by reducing the nitrate levels in the water. Our second objective is to assess the capability of Magnolia, which is used in artificial floating islands. Next slide. So the question arises, what is an AFI and why an AFI? An AFI is a human-made platform designed to float on the surface of water bodies. They are usually made out of PVC pipes or other floating material. AFIs have various benefits, such as reducing nutrients in the water like nitrogen and phosphorus. They also regulate the amount of sunlight that enters the water. AFIs increase the biodiversity in lakes by allowing birds to lay their nests on them and allowing tadpoles to take shelter in their reefs. AFIs are also immune to fluctuating water levels. This means that they can be implemented in almost any water body. AFIs are also low cost and easy to implement. Next.
So our study area for this project is Putnali Lake. Its perimeter was 927.3 meters. Area was 55,158.65 meters square. There is even a QR code that will directly lead into YouTube for the more better clarification of the scheme.
This is a timeline of Putnali Lake. In the year 2009, BBMP started the rejuvenation of the lake. In 2023, the lake got covered in weeds as rainwater got filled into the lake. Deweeding was also carried out in this lake. In 2016, AFIs were introduced and testing and analysis of the water sample was conducted. In 2018, sewage from the adjacent layout led to a massive fish kill.
2019, a drain connected directly into the lake. This undid all their work. In 2021, BBMP desilted the lake, deweeding was also carried out, and the fountain was reinstated. In 2023, 7 AFIs were added. They were in the dimension 5 feet into 5 feet, and the lake now is in currently good condition. Next slide. This slide shows the nitrate levels in Puttenhalli Lake. As you can see, the nitrate levels have had a considerable fall from 2016 to 2024. This reduction of nitrate level shows that the water quality has improved. Next slide. To assess the capability of colocasia plant, which is used in AFIs, we have done this experiment. We took a tub and filled it with 20 liters of tap water and placed 5 colocasia plants in a floating lake. We maintained a control which was water without the colocasia plants and tracked the nitrate level for a duration of 10 years. The initial nitrate concentration was 20 ppm. To test the nitrates, we used a commercially available nitrate testing kit. This kit works by adding a reagent to the water sample. The reagent reacts with the nitrates in the water and produces the color. The intensity of this color is compared to the color chart which is provided. These are our results in observation. The initial nitrate level was 20 ppm at the start of the experiment, and the final nitrate level was reduced to a range of 0 to 5 ppm. The control that we maintained consistently showed 20 ppm throughout. Our observation here is that colocasia plants in use in AFIs are effective in reducing the nitrate levels in the water. Next slide. These are the additional conservation efforts that are done at Puttenhalli. PVC pipes which are used to make the AFIs are replaced with bamboo ladders to reduce the microplastic contamination done in the lake. When we went to Puttenhalli Lake, we spoke to the gardeners. They told us they clean the AFIs regularly. A fountain has also been installed in Puttenhalli Lake to aerate the water and address low oxygen levels. Dry leaves are used along with cow dung to make compost in Puttenhalli Lake as well. Various trees have also been planted around the lake area. This forms a green canopy around the lake. Next slide. These are the conclusions. The colocasia plants that are used in AFIs are effective in reducing the nitrate levels in the water. The effect on colocasia might have an effect on other water quality parameters such as pH, water quality, turbidity, hardness. These should also be tested. And we should also explore other combinatory methods like bioaugmentation along with the AFS for maximum benefits. This is a picture gallery. As you can see in this image, the lake is now in good condition. We noted that many people were walking around the lake and there were many signs of bird watch as well. Next slide. We extend our heartfelt thanks to our institution, AECS Magnolia Maaruti Public School, and our teachers. We would also like to express our gratitude to Dr. Vinay Kumar, Mrs. Usha Rajgopalan, Mrs. Divya Shetty, and Mr. Nataraj Pandit for their invaluable guidance throughout this project. We would also like to thank the Indian Institute of Science for their invaluable support. We would like to thank the Indian Institute for providing us with sponsorship. Next. The references are listed below. Thank you.
Two slides on this one, you should read the literatures and write about the floating wetlands. What are the species commonly used in India and that too in Bangalore, and where are the successful things? There are many lakes have that floating wetlands, okay? Epagodu and such, this one. You should also say that what kind of plant they have planted and what is this, it's all reported in the literature. You bring the literature.
Colocasia plants are used and also canna plants.
No, I agree with you in Puttenhalli, but there are other plants also used. So that's where you have to do the review of literature, say that, okay, in this lake, these are the floating plant, the floating wetland, and these are the species used. And if there is the experiment people have done to show what is the improvement has happened, like you have shown, that would be good, okay?
Yes, sir.
Actually, your work is different work, okay? That's why it is good that you are using the 8th standard also. So I found the thing that your heading is that artificial floating lines, okay? So you should give the WhatsApp owner that
wanted to select colocasia because this plant was easily available for us. And we also found that these plants were used in putinari lake. So we wanted to mimic the conditions in that, in the lake.
So in that floating island, that colocation was there. That's what
Yes, ma'am. Yes, ma'am.
Okay, then another thing, then why that reduction happened, that nitrate value, given the 2016 and 2024, that higher reduction. What type of conservation activity they did? Then for what are the activities like that the reduction is happening? You just mentioned about that part. Okay, in that study that you have given the graph now, there itself you are telling like the reduction 2016 to 2020. Then what are the methods? Because you are given the acknowledgement to Usha and Magnolia, you can go and ask them what are the activities they did for the protection for that potentially. That also you should mention.
See, actually, 2016 the sewage was entering the Putinali lake and it was stopped. So that is where you should understand what she is saying. Why the changes have happened. One could be because of floating wetland. The second is the diverting the sewage. They have diverted the sewage. You have to probe that further. You talk to Usha and Vinay Kumar also. They will tell you about that. And then do the experiment properly. Huh? Yes, Silva.
so I feel that you have to do more experiments. Okay, check that the house colocation is removing the odd parameters. Then you have to do some more days of experiment and then do the water clarity. Okay.
Yes, ma'am.
So in that you have I didn't see the final. You can also do chloride, calcium, magnesium apart from those parameters which you have written. Okay. Then it's like the reduction in the parameters means it's called bioremediation. So you can also talk about bioremediation in one slide. Bioremediation by these floating plants. Okay.
Yes, ma'am.
Okay. Thank you.
I am Arjun Bhattacharya.
I am Pratham.
I am Rudra. Today, we are here to present you about our topic, Ecosystem Diversity Value. Our author recommendations are AECS Magnolia Maaruti Public School. Before we delve into the project, let us see what our objectives are. Our objectives are to study the ecosystem diversity around the lakes, to understand and appreciate the importance of reviving and preserving the lakes in the ecosystem diversity, to know the interdependence of the elements of the ecosystem and that if one species declines, the others can take place. The last, to learn about the role of waste management and environmental awareness in preserving the lake's ecosystem. Next. Next. Let us see what our research methodology was. The primary research was that the team members went to the Putinali Lake and explored the ecosystem around it. The secondary research was we referred to the various websites and the internets, the details of which are given at the end. Let us start. What is ecosystem diversity and why is it important? Ecosystem diversity is the variety of habitats found in a geographic location and the variety of species it inhabits. It is a type of biodiversity that refers to the variety of communities, ecological processes and the beneficial services found in a geographic location. It is very essential for the survival of all species, including humans, because it makes the ecosystem stable and resilient to change or extinction. Without ecosystem diversity, the ecosystems become more vulnerable to severe change or extinction. Next. Now let us see the value of ecosystem diversity for the environment. Balanced food webs. A diverse range of plants and animals creates a balanced food webs. The predators keep the prey population in check and the herbivores maintain the population of the plant growth. It enhances resilience to changes. That is, if one one species declines, the other species can take over, hence making it more balanced. There is also nutrient cycling. That means the nutrients that come from plants to the animals go back to the soil through decomposition by the bacteria. It also provides ecosystem services such as provisions of essential services like clean water, pollination, climate regulation, etc. This is our study area, that is Puttanhalli Lake, Bangalore. Let us delve into the case study. The PNLIT, that is the Puttanhalli Neighborhood Lake Improvement Trust, arose out of a worry among a very few residents on the plight of the extincting Puttanhalli Lake, also known as Puttanhalli Arekere in JB Nagar, that is South Bangalore. Through their dedication and innovation, the PNLIT has transformed the Puttanhalli Lake into a thriving alpine sanctuary and a vibrant ecological hub for the neighborhood. This lake has a variety of species of birds, trees and plants, hence creating ecosystem diversity. A flourishing green haven has been made around the lake's edge. The walkway is a great place for everyday jogs and walks. People have also adopted some trees and blessed for their survival. Here is a table of the trees spotted in the lake. On all the trees there was a board with their name, biological name and family too. We spotted many trees like Gulmohar, clusterfig, temple tree, Nile tulip, India beech tree, wood apple, etc. Here are the pictures of those boards from which we got the information. There is also a sustainable water source which uses the treated wastewater from a nearby apartment complex. This is the first sustainable water source for Bengaluru and a pilot project for the Karnataka State Pollution Control Board, that is the KSPCB. There is also an enhanced habitat for birds. Apart from the planting perches, an aerating fountain has also been installed. Many species of birds have been spotted, like red-watered lapwing, grey-headed myna, common myna, among the other migratory birds. We also spotted birds from which we got to know that the migratory birds and the other birds will come to the Puttanhalli Lake. Next. There is also cutting-edge floating biofiltration platforms to further purify the lake's water, and these biofiltration platforms have been inspiring similar efforts in other lakes too. The leaf compost bins are also seen along the path. There are various bins for making compost from the leaves, which are installed along the lake's walkway, which are regularly cleared. The lake is also exempted from commercial fishing, and the fishing has also been banned, which has safeguarded the ecosystem. It has also preserved the ecology of the fishes. The litter around the lakeside is also visible, though the litter bins have been installed at regular intervals. To conclude, I would say that Puttanhalli Lake is a perfect example of how a lake can be revived into a beautiful water body. Numerous trees have been planted and people can adopt them too. A variety of birds have been spotted. Volunteers and residents continue to help in the beautification of the lake. Effective waste management is also visible. Despite the efforts of PNLIT, the owners lies with the villagers who should ensure not to litter around and maintain the lake's beauty. Acknowledgements. We would like to thank Dr. TVRAMACHANDRA sir for explaining us about the ecosystem diversity and giving us the curiosity about it. Dr. Seema Goyal ma'am for giving us the opportunity to participate in this event. Divya ma'am for guiding us for the presentation. Our parents who were one of the main support systems helping us to achieve the outcome of this project. Here are the references from which we collected some information.
What is the role of two of you?
So,
You are standing like Dwarpalaka only. Your name is also Dwarpalaka.
Sir, he had prepared the slide, so we thought of telling from the slides. So he had to only tell the whole thing.
No, no, you should divide, okay?
Yes.
You, one of you do the results, other one introduce like that. It should be collective effort. Huh? See, like I gave a suggestion, no? What you have to do is, you need to have 2 slides on literature review. There are many studies people have worked on, put in only, okay? And also there is something called Bangalore Lake Information System. You can also see that, okay? It is also called BLIS, B-L-I-S, okay? See.
I have some suggestions regarding your heading first. Ecosystem diversity value. Okay. That ecosystem, then your heading is talk about the ecosystem diversity value. But we have different types of ecosystem. So you are telling about the wetland ecosystem. Then that you should be changing the heading. Okay. Another thing, the Putenahari lake, the diversity value you are telling, no? There is you are given only 3 species listed. There is bird species also it is there. Then macrophytes also is there. Other small grasses or different things are there. Now that you should include that one also. One science. Okay. Then that family, that which tree is the higher family is there. That also you can tell now. Then which one that medicinal value is there now, that because you are given the value, the ecosystem diversity value. That means you should read the some, there is fodder collection is there now, then how much amount of fodder they are collecting or anything, that then fishing is there now, yeah, fishing activity, then how much fish they are getting, then what is the value, that also you should be, okay, then it will be very good.
But the fishing was banned because the fishes were dying, so they banned the fishing in the lake, hence the preserving the ecology of the fishes.
No, what kind of fishes are there, you can list that, okay? You can talk to the people, that's where I said, there are published materials are available. From that also you can take and write. And if there are some people who know about fish, you can talk to them. Sulabha, you have to keep in mind they are 8th standard. No, that is where you have to keep that, okay? They are too young actually. Wait, your classmate is here.
You have prepared for trees, right? You know all the trees, you have seen. So like Magnolia and all, you know the Kannada name also of them, right? Or you can ask your parents or your grandparents, Kannada names. Okay, you can add that in this slide. That will be very good. Okay, and then one more thing. Each tree, like gulmohar tree, how many gulmohar trees you found out in that area? That also you can write. Yes. Number of gulmohar trees in that lake, or number of mango trees in that area. That also you can add. Okay. Then one more thing. The next slide you go. There the name of the birds you have written, right? That also you can prepare a table like this. So that will be more prominent
change the title.
A very good morning to my presenter. My name is Arun.
My name is Suryansh.
And my name is Palak Kumar. We are students of 9th grade of AECS Magnolia Maaruti Public School, and today I want to present our research on the comparison of bird biodiversity in 3 different areas.
We are here ornithologists who have been interested in nature and have admired ourselves toward its beauty since childhood. So today we have a case study where we compare the biodiversity of the birds in 3 different areas.
The first area is Kodichiknahalli in the Bengaluru South District. It's a completely urban area with very mild vegetation.
The next area is the Madhiwala Lake. This region has an area of about 114.3 hectares, situated in the Bengaluru South District, and has a very few macrophytes and has a short and bushy trees and plants.
The third region is the Ramanashi Nagar, which is situated in the Kamannahalli main road. This has less vegetation and has non-flowering and non-bearing trees.
Our main objective for this study is to observe the biodiversity of the birds in the 3 different areas and also observe the possible factors affecting the varied bird population.
The apparatus used in this case study are notebooks, pen and pencil, camera and iNaturalist website, followed by binoculars and cell phone.
The duration of the study was 32 days, which was from June 1st to July 2nd, and the timings were 6 am to 7:30 am in the morning, followed by 5 pm to 6:30 pm in the evening.
So this is the tabulated data of the birds observed in Kodichiknahalli. So as you can see, the majority of the birds are from rock pigeon, Indian jungle crow, house crow, common myna, Asian goels, grey pigeons, etc. A total of 15 species were observed.
This is the tabulated data of Ramanashi Nagar, which has less vegetation, in which the top 10 birds are blue rock pigeon, red-whiskered bulbul, jungle crow, rose-ringed parakeet, common myna, Indian spotted eagle, house crow, black kite, eastern imperial, black-throated monkey.
And now finally, we have a tabulated data of the Madhiwala Lake. This region has a total of 37 species, ranging from bulbul to the spot-billed pelican. So the most common species found in this area are bulbul, pigeon, peacock, myna, cranes, Indian spotted eagles, crow, painted stork, kingfisher, and lastly the purple heron.
So this is the overall recorded data of all 3 areas. As you can see, there is a constant pattern that the most species are observed in the Madhiwala Lake and the least in the Kodichiknahalli area.
So this is the comparison of the totally collected data, where we can see in Madhiwala Lake the number of species observed are 27, whereas in Ramanashi Nagar the number of species observed are 24, and in Kodichiknahalli the number of species observed are 50.
And yeah, this is the final chart for comparing the number of species observed. So here we can see that the number of species in 3 different areas is directly proportional to the amount of vegetation in these areas. So the highest number of vegetation is in Madhiwala Lake because the total number of species is 37. The intermediate number of species is 24 in Ramanashi Nagar, followed by Kodichiknahalli, which has the least number of species, that is 15.
So this is the diet of the various bird species observed in the Kodichiknahalli area. So as you can see, majority of the bird species are omnivore, followed by carnivore, and the least are from piscivore and herbivores.
So this is the diet species of bird in Ramanashi Nagar, while most of the birds are carnivore and omnivore, and the least are piscivore and herbivores.
And now we finally move on to the diet of the bird species in Madhiwala Lake. So there are total number of 37 species. Out of these, the majority of the bird species belong to the carnivores and omnivores, whereas the least number of species belong to the herbivores.
So these are some of our hypotheses observations to find out why the results which we have observed. So in urban areas there is very low vegetation, so not many trees and edible plants for the herbivores and frugivores, so they cannot usually thrive in the urban area. So Small insects and worms are very common, so it is good for the insectivorous species. And in urban areas, there are many people who might feed grains to birds like pigeons, so their populations can drastically increase. And human activities like deforestation and habitat destruction are causing a decline in population of many bird species.
Some observations on why carnivores and omnivores are mostly found in the semi-urban areas are as follows. Abundant prey. Semi-urban areas often have a mix of natural habitats and human settlements, providing a diverse range of prey. Edge effect. These areas often have a mix of open spaces, forests and gardens, creating edge habitats and attracting insectivorous plants. Reduced predation. Predation pressure from larger predators like big cats or wolves is generally low in such areas. Human activity and adaptability. Many carnivores and insectivorous birds are adaptable to human-altered nature. Also, there aren't many water bodies for fishermen.
And now, let's see the observations on why do the birds have such diets in natural habitats. First one, the food for omnivores and carnivores can either be in the form of aquatic animals or the insects that are situated near the lake banks, so they receive sufficient amount of water which helps to sustain their thirst. Herbivorous animals need a longer digestive system to utilize the plant as the food resources efficiently, hence it is difficult to find such birds in nature. The areas nearby the lakes have a huge amount of vegetation due to the abundant availability of food and water, attracting the few existent herbivores and frugivores. The mild human interference in this area has reduced the number of predators which can hunt these birds with different diets. Many small omnivorous birds aren't eaten by the large carnivores as they can easily blend in the thick and bushy leaves, due to which they are inexpert.
Why does bird population decrease as we move towards urban areas? Trees are being cut constantly, thus the habitats of birds are being destroyed, and birds are migrating from this urban area. The lack of vegetation in urban area destroying the natural habitats. Many lakes are being dried up to make apartments, buildings, etc. The high rate of urbanization is leading to lack of prey for many carnivorous and omnivorous birds. These are the basic conditions causing the decline in bird population.
Factors affecting bird population. Some of the main factors are weather, habitat loss, air pollution, predation and competition, disease and parasites.
Now, let us see the steps to increase the bird population in Bangalore. These are the following steps. Encourage organic farming practices and limit chemical pesticides usage in urban areas. Install bird houses and nesting boxes in suitable locations. Educate the public about the bird conservation and foster appreciation for the urban biodiversity. Maintain clean water resources like the bird baths. And lastly, minimize the collisions with the buildings through the design solutions.
These are some of the fun facts about the bird which we found while doing the research. Birds usually stand on a single live wire, so electric current doesn't flow through the bird, hence they don't get shocked. There are over 10,000 species of birds all over the world. Only 2% of these are herbivores. There are about 570 species of birds in Karnataka, 340 in Bangalore, in 1980s, which is declining due to urbanization. Birds evolved from a group of meat-eating dinosaurs called theropods, although they evolved from the small theropods. All birds are classified as members of the kingdom Animalia, phylum and class Apes. The characteristics that all birds share are that they are vertebrates, that is, that they have a backbone, which is what makes it an animal of the phylum Chordata.
This is the picture gallery showing the pictures collected during the study. Our experience. Overall, it was a delightful experience. Bird watching became a part of our routine. On the first few days of observation, it was observed that most common species were the blue rock pigeon, common myna, house crow, etc. But as the weather conditions became better, birds began appearing in great numbers. We enjoyed collecting data, traveling to the lake every day, clicking pictures and later discussing which birds I had seen that day. We were able to appreciate the secret heroes of the ecosystem, the birds. All of us have fallen in love with birds, their beauty and fascinating powers and skills.
We would like to express our gratitude to our respected principal, Dr. Seema Gure ma'am, Vice Principal Jayashree ma'am, Gayatri ma'am, Ishwari ma'am, Divya ma'am, and expert Vijayal sir, and all of the members of IISC for giving us this wonderful opportunity. These are the references and websites we have used to complete this presentation and research.
Hope you all have enjoyed and will help us on our journey to protect the bird kingdom. Let's all be ready for a world filled with harmony between mankind and nature on our wonderful planet. Thank you.
You know, you need to have one or two slides on birds of Bangalore. How many species you listed that in the last? So initially, if you can, and also if you can talk about generalists and all those things. You know, various groups you had. No, what do you mean by that? Okay. The third thing, which is more important, is the conservation status of the birds. You know, endemic, endangered, that kind of a thing. No, you critically endangered, all those things. Then you can say that how many endemic species are in a rural lake and how many are generalists or a thing. You know, comparison you can say that. That will also reveal ecological status of the region. That's very good. And since you people have worked on the birds, probably you should have continued for more time. You know, it gives you a lot of this one, you know, satisfaction, and there was a smile on your face when I was seeing you all. That may be because you work with birds. And if you want that to be continued, continue that journey. Every Saturday, Sunday, you try to do that, okay? It's always good to do that.
So you also continue to study for the different seasons, right?
That's what I meant, you know. You should continue the thing. And this would be very good actually. If you can enrich that with the photographs and the, you know, when you go for the field visit, you can. And also you can talk to some people. What do they feel about the thing? One slide you can say that public opinion about the ecosystem. You know, the purpose of this symposium is to make more and more people aware of the ecosystem and why it is required. You know, this kind of experiment will help, okay? Yeah.
Actually, your study is that both sides. In your face, that your smile is there, but you wanted to give that smile to audience also. So you should give that in the introduction part. Why you are selecting this birds subject? You have plants also there, other subject is there, but you selected birds. That means something interesting is there. Huh? You should bring that. Okay, then ecological importance. Then they were giving the different services also. That part you should do. Then you have given the carnivores, then herbivores and all the things. Now, then some students they don't know. They are not understand the. So you should in the introduction part you should whatever you want to tell. You give brief small explanation about it. Then that list part also it is there. That the difference in Manipala and all the things. Then the comparative type, the Manipala, that graph together, that three things together, then it will be very easy to interpret. Okay, you can also will control that slides also. Then it will be that list you can give the separate, but the herbivores and the carnivores, these are the things that you graph, percentage graph you are given. That you should give together in one slide. Then it will be interpretation part is very easy. Then you should tell why carnivorous species are higher.
Because there are more number of piscivores and insects found in the Maniwala lake. So majority of the species we find near the
And
lake
food availability
is also more in that
that
area.
means why that the fruity
Frugivores,
frugivores,
they are they are comparatively less.
less. Why then you should bring that one more.
Okay, reason.
Yeah. That's one of the reasons of the you should bring that. Then herbivores also that is there. Then aquatic that you are telling about the Manipala lake near the thing. Then aquatic plant and then birds also is there now. That also you just birds. And aquatic plants and birds. That you should that one slide you should bring that what are the things you can list out and
The lakes you should mark by polygon, not point. See here you have marked, it is not very clear. You should draw a polygon around the lake so that you will also get to know the area of the lake. Okay? Okay, and one more thing, in the list, if you can add the Kannada names also.
We added one that.
You added.
But you all from some other place, no?
Yeah,
You are from?
Rajasthan.
Huh?
Bihar. Bihar.
Okay, so you can add minerals also,
Yeah, sure.
if possible.
Yeah, sure.
Then one more thing, you have shown a funnel chart of comparison of the number of trees and birds. But in that chart, the number of trees is missing. Hannah?
But we are talking about the bird
Yeah, I
biodiversity.
know.
we-
So I have one suggestion. If you can do, in each lake, how many trees are there? Okay, now you have the number of trees and number of birds. So then you can do a this heading will be appropriate. You understood what I am trying to say?
Yes, ma'am.
Then you can say that this is why the number of birds are less. Okay, that's it. Thank you.
Thank you, ma'am.
Okay, thank you.
Jai Guru Dev and good morning to one and all present here. Myself, Lily. Today we are going to talk about the importance of butterfly in our ecosystem. So first we are going to talk about what is the difference between butterflies and birds. Why don't we call butterflies and birds as butterflies? So the major difference between butterflies and birds are that birds have bones whereas butterflies don't have any bones. Birds are non-vegetarian whereas butterflies are vegetarian.
Butterflies help us understanding the biodiversity, the ecosystem, and the environmental health, and even decreases the population. My objectives are the identification of different species of butterflies and to inform everyone about their habitat.
So the methodology for this presentation is that we visited our garden between 5 to 6:30 a.m. The places which we visited were Arekere, Begu Lake, and SNL Ratsarani apartment. The duration for this presentation is 32 days and the materials required are book, notebook, camera, pen and pencil.
What are butterflies? Jewels, the flying jewels of nature. They play a very crucial role in the ecosystem as pollinators, contributing to the reproduction of the plant and biodiversity. They plays a very important role in the maintenance of the nature, and it belongs to the species called Lepidoptera.
So now going further with the study area. So the first study area is SNL Ratsarani, which is located near Gopal Road.
Try to finish our.
So the next study area is Begu Lake. And the last study area which we are going to talk about is Arekere Lake, which is located near Peria. So now let's go deep into some butterflies. So the first butterfly which we are going to talk about is common glass yellow. It is from Maaruti species. It is generally a very light yellow color species and they are adaptable to various environments. The next butterfly which we are going to talk about is common tiger. It is from Danus genitiva species. It is generally known for its striking appearance and its population can be decreased due to pesticides and it plays a very crucial role in our maintaining our environment. So the next butterfly which we are going to talk about is common wasp. It is from Papillopolita species. It is generally a large color butterfly with black and white striking appearance pattern. Its population can be decreased affected by habitat. The next butterfly which we are going to talk about is blue tiger. It is from Tirumana liminal species. It is generally a large dark black color butterfly with blue streaks on its wing, and these are known for migrating over long distances and its population can be decreased by pesticide use. So now we are going to talk about the record of the study area 1. So in study area 1 the duration which was, so we went to the places between 5 to 6 a.m. and the 30 minutes we wrote about the butterflies. Now, so we can see that the most butterflies seen was common warmer, which is 22%, and the least was blue tiger, which is 11% in SNL Ratsarani apartment. here also we got 6 butterflies we researched about, and so the record for Begu Lake is that the most butterflies again were plain tide, which is 29%, and the least was common normal, which is 15%. We saw the decrease between from Asan and salinity to Begu Lake. So here is the comparison between SNN Lake and Begu Lake, which we visited on the same day. So over here we can see that there is an increase in common grass yellow butterfly in Begu Lake, and whereas there is a decrease in lemon different butterfly by 19%. Now we are going to talk about the record of AECS, which was Arekere Lake, and we visited this place from 18 June to 3 July. And the record for this number of species in Arekere Lake is we saw that lemon different, which was 21%, and the least was common normal, which is 13%.
Talking about the host and metro plant, the common title, the host plant is scarlet and the metro plant is antenna. The common model, the host plant is antenna and the metro plant is citrus trees. And the question mark, the host plant is hackberry tree and the metro plant is butterfly bush. And the red bamboo, the host plant is necklace and the metro plant is marigold.
Going forward by the measures adopted, so the first measure adopted that we provided water sources. So we filled water in some bucket and kept it outside our windows. So we saw that many butterflies and birds were coming and drinking water from it. And the second thing which we did was we planted many native plants such as marigold and antenna.
Planting more nectar plants leads to more butterflies, and more butterflies leads to a very good environment. We started observing the actions of the butterflies, and we understood how important butterflies are to our environment. In conclusion, nectar plants attract more butterflies, and most common butterfly species was European bird, and it helps us understand the importance of the butterflies and recognizing the various threats to the butterfly population.
So the acknowledgement we would like to thank everyone who has supported us, especially our parents, our principal Dr. Sina Goyalma, our teachers, Gayatri ma'am, Monalisa ma'am, and Ishwari ma'am, and our friends who have supported us. And here are some questions we would like to answer. The first one is how to conserve butterfly diversity. So if you have 1 hour per day, it's enough to get familiar with butterflies and count where the butterflies are located. Where are butterflies found? So butterflies are found all over the world in all the types of environments such as warm, cold, rigid. It is mostly found in the tropical region, especially in tropical rainforests.
Europe and Magnolia are species of butterflies recorded in the city. And
What is the most famous place for a sight of butterflies in Bangalore? If you want to get more familiar with butterflies, it's suggested to visit Arekere, Bangalore, Nashika. It's over there you can get familiar with more butterflies and understand their nature and habits. Thank you.
What you have to do is why butterflies are required. In the initial slide, if you can highlight that, especially the pollination and why pollination is required, food. That's one thing. Second thing is there are couple of slides about each species you talked about. Now, can you put it in one slide in a tabular column? Okay, that would be better so that it would be convenient for you to explain also. And also you should do literature review to say how many species are there in Bangalore and the various their ecological significance of the butterflies. Okay, so then that will help. And also how many migratory species come to Bangalore. That also from the literature you can put in. There are in our portal Chaturved presentation is there, and also the book is available. Please go through that. Okay, it's a good job. Okay, yeah, since you.
One suggestion that you are talking about the butterflies now. When a butterfly's life cycle,
mic.
5 to 6:30 a.m. and now we have changed the time. Now we are measuring in evening so that we get to know about the butterflies and what model.
Yeah, you should tell why that is the thing. Yeah. Because you only know the butterflies. We don't know anything. You will teach us now. That's why that is the confidence. No? So you should tell the what is the behavior thing and all. You just give it. Then this there are also some reasons. That is also you to give in your okay. Because you are in the 10th standard. You should be more scientific.
Is that your species name or not in the
So, in the beginning, right now we are using birds as vegetarian, non-vegetarian. Some birds are frugivores also.
It's like very less, but comparatively it's like very less because most people are afraid of vegetarian. Butterflies are generally vegetarian because they go from like they take food from their like
That is okay, but don't make them comment that birds are vegetarian. Okay. So, and one more thing I have to you have put the objectives in your first. So, first your introduction should come as sir said, you know how butterflies are communicating, all those things, and then your objective will come. Okay? And the study area maps that you have prepared, the right area and the surrounding area that you have surveyed. You should mark that area properly and you should mark how much area you have surveyed. Okay? That's
Gentlemen, good morning. My name is Sandhya.
My name is Shresh.
So today I will be opening the presentation for study on the topic socialism in plants. So everyone usually thinks that plants cannot feel, see, hear or talk, but we believe otherwise, that life as it exists has consciousness, and whether it is an animal, a plant, a microorganism, or anything. So we want to prove our beliefs to the scientific world, and that brings us to our project. So it is a small experiment requiring only a few fenugreek seeds, a few pots, and next slide.
Siksana had planted fenugreek seeds in 5 pots. In the first big pot, she had sprinkled 30 seeds and only 1 seed each in the other 4 smaller pots. Sanmay had taken 2 pots and she had sprinkled 30 seeds in each of them, but had kept them in separate groups. She was tasked to sing to one of the pots and to keep the other one alone in similar conditions. For all the plants, the same soil was used and they were given an equal and measured amount of water. All plants were kept in similar conditions, but one aspect, company of other plants or other humans.
So as Shrey said, plants were kept in isolation, in groups, and with human interaction for 14 days in their residence, and these are the results.
In the first experiment, we can see that there are 2 groups. One group are plants that were grown in groups and the others were grown individually. You can see that the seeds that were grown individually grew at a much slower rate, and compared to the seeds that were grown in groups, they sprouted much later and at different times. By the day 14, when the first group had grown to its maximum rate, you can see that 2 of the seeds in the other group had not been sprouted yet. In the next experiment, we can see that Sanmay has come to the plants, the pictures are on top, and the ones that were kept alone are on the bottom. We can see that the ones that she has sung to have also grown to be much taller than the ones on the bottom, more than 2 times their height. There is a big difference in growth based on whether or not you have been exposed to other plants or human voices.
So this is a graph that shows the difference in the heights. So as you can see, by the day 14, the height of the seed sowing group reached up to 8.5 centimeters. While the height of those who were sown individually was just 2 centimeters. So the height of the plants that were sown in group is 4 times the size of
growth, the height of the plants is affected by whether or not there are other plants or animals near them. So we've spoken to a few agriculturalists, and here are their opinions.
While most of these agriculturalists had neutral opinions about the project, we can say that Mr. P. Shankar Bhatt said that plants, when they're grown individually, will grow way better than plants when they are grown in groups. Whereas Mr. Suresh Gowda completely agrees with that point. He has said that Mr. P. Shankar Bhatt says that plants look out for one another by working together, recycling nutrients and supporting each other. But bunching too many seeds in one place will leads to competition between plants and they will not grow very well. Whereas Mr. Suresh Gowda says that plants complement each other and maximize their growth, and humans speaking to plants makes a big difference in the growth of the plants. Mr. Narahari Prabhu from Peraro village in Magnolia says that integrated farming blends crops, animals and fish to support each other, while organic farming uses natural methods to keep soil and plants healthy. He believes that plants need to share their habitat with other plants or animals to grow properly. Mr. Thirumaleswar Bhatt from Kodiyadu village in Siddhya Taluk says that plants grown in a single pot will fight for nutrition and hence won't grow properly. And that while there might be a possibility of plants growing better in the presence of animals, it will have not much of a difference.
So Mr. Narayan Madhurite from Arekere in Kota thinks that plants require human interaction to grow properly, and spending time with plants is beneficial enough for the plants and humans. So Mr. Narayan Madhurite has implemented organic farming methods in many fields and has earned many prestigious awards. Next. So we know that plants grow better when they have human interaction, and they react to their environmental stimulus through physical or chemical manners. For example, when an insect feeds on the leaves of a plant releases jasmonic acid, which is a hormone, to either kill or shoo away the insect. And if you touch Mimosa pudica, the touchstone of plants, the leaves close and they drool, showing the rotting of the plants, which will deter the herbivores from eating them.
We want to prove that while there are negative reactions of plants to environmental stimulus, there might be positive reactions as well. For this, to broaden our study, we have planted both jasmine and wheat seeds on 12th August, that is yesterday. And we hope to prove our theory by broadening our study.
Thank you.
One of the things you have to do is enrich the content with the review of literature. Okay? Because some of this is a new thing what you talked about it, but you said socialism first. That's what you mentioned. Socialism is different from social organisms. These two are two different, okay? Unless you understand the terms, don't use it like that, okay? And the second thing is when you do the experiment, it has to have the similar kind of the environment. Suppose if you are putting the individually, if they are with the humans, that same this one you have to maintain. And you include the people. That the slide you named it as conference. That is not conference. That is interviewing this one, no? That you have to reword it, okay? But the topic is interesting. That I agree with you. So when you talk to the plants, plants will give you the fruit. If you don't speak, it won't give. So those aspects is well documented. Okay? Maybe after the question I will tell you about that, okay? Sincere.
Actually, the building is planned social programs. Okay. Then let's come to the outside that is sites and all. Then another you have given the plant growth. Then seeds and like size. But you didn't mention what is the one seeds.
Because the others are gone for food.
that's why please do it a lot of literature. But because in these slides we never stated what you wanted to do. Because you have given me some interviews and all the things, but it is not coming to the effective. Okay, because that you are telling that there are plants social organisms, that plants, that leaves has also will tell the story. Okay, you know that the leaves cover and all the things, the plant leaves cover, you study right? Everything you should bring the thing. Then you can bring the our medicinal thing, how they help the plants. Maybe the plant, the medicinal value also you can bring. Then in our Veda and all, lot of their importance. Then you, I think you are listening to our staff's topic also that he is telling about the importance of plant, you know. That all the things should you please bring some more data. Because you are in 10th standard presentation. Please do it properly. Okay. Don't lose your mark or something. So do it some more. Now your presentation is over. You will get some more value. Please catch the lot of data and arrange it as a properly and present it properly. Okay.
Okay, Cincy, next. Okay, you have anything?
Graphs and showroom comparative two slides you have put two graphs, right? One is with social interaction, one is without social interaction. You can put the graphs in you can prepare a single graph and put those in a two different colors. Then the comparison will come better. Like that, with literature that sir and ma'am suggested, you should implement that and enrich the data.
Jai Gurudev and good morning. My name is Monika and today I and my teammates Danika and Pranav are here to present you about a comparative study of clean and polluted lakes. So as we all know that lakes play a vital role in our environment and are also a way of sustainable living for the humans. So first, starting with the introduction. Clean lakes support diverse aquatic life and are safer for the human use.
Hold the mic properly, it is not audible.
Okay, so the clean lakes provide support this diverse aquatic life and also are safe for the human use. They also provide a better biodiversity. The polluted lakes contain toxic contaminants which reduces the biodiversity and also negatively impacts the ecosystem. So today, studying the lake pollution helps us identify the contamination sources and develop strategies for sustainable water quality. Our objectives today are to compare soil quality between the Madiwala Lake and the Arekere Lake, to study the biodiversity of birds, to identify the commonly found macrophytes, and also survey analysis of the local lakes. So our study area for the clean lake was the Madiwala Lake. Its location is Bangalore South between the Baneganda Road and the Hosur Road. Its area is approx 1.113 square kilometers. Our study area for the polluted lake was the Arekere Lake. Its location is Bangalore South, close to the Baneganda Road. Area 0.519 square kilometers. So today we have also done a soil analysis using the NPK kits, and we have done it at our nodes. We have done it on different parameters like temperature, pH, nitrogen, phosphorus, and potassium in kg per acre. For the as you can see, there are the numbers vary in all the parameters for both Madiwala Lake and Arekere Lake. The nitrogen
here shows that it's better in Madiwala Lake as it helps the plants and the algae growing over there as and also does the nitrogen fixation underwater. Next, the phosphorus levels are also different because at times due to the runoffs of the agricultural waste like fertilizers, harmful pesticides, and also sometimes the factory waste, the numbers vary. Then last, potassium. The potassium also has the same reason because more of the factory waste, fertilizers, and more of agricultural waste gets dumped into the lakes.
So these were the predominant species of birds which are found in Madiwala Lake as well as Arekere Lake. Our sample of Madiwala Lake is the lake. So first one we found the Jawan Pondera, the scientific name for Ariola spinosa. It is found in shallow, fresh as well as saltwater wetlands. They usually feed on insects, fish and crabs, and they are known as predators who help to control the smaller species numbers, thereby preventing their growth to the land. The second one being the Little scientific name for Arekere garzetta. They are found in both saltwater as well as freshwater wetlands, and they mainly feed on fish, amphibians, reptiles, etc. And they feed on the ectoparasites which are present in the marine animals' bodies, which serves as a source of nutrition for them, and the marine animals in turn are happy to get rid of the parasites which are present, thereby forming a symbiotic relationship with each other. The third one being the dowdard, scientific name for Anhinga anhinga. They are found in creeks, river pools as well as shallow and deep lakes. The diet, they usually feed on mining crustaceans. Crustaceans are nothing but they are lobsters, shrimps, crabs, etc. And next one is the Arekere lake, which commonly name is Eurasian food. Scientific name for Fulica atra. They are well adapted to urban environments and they are usually omnivorous, which feed on live prey, eggs, etc. Commonly, dark-capped beetles, scientific name for Magnolia tricolor, they are found in and around the human settlements and they feed on fruit, petals, nectars, seeds, etc. And they serve as pollinators, which help in transferring the pollen grains, thereby aiding the reproductive process of the flowering plants. We also visited Dorakalsanda Lake where we found the female peacock, the male in its swarm pen, and the male peacock as well as the captive pair. Now, as you when we compare these species which are found in both the lakes, we can see that those species which are found in Arekere Lake are well adapted to urban environments because they are versatile feeders. They have a wide variety of food sources which they can feed on as compared to those which are found in Madiwala Lake, which usually feeds on insects, etc.
So I'll be briefing about the macrophytes that we observed in both Madiwala and Arekere Lake. At first in Madiwala Lake, we observed the Indian snot shark, which is mostly used for treatment of industrial waste, and it is rich in starch and poses as common fodder for most of the minute organisms present in the lake. Next is the knotweed, which is a flowering plant and which has many medicinal benefits. It is also these are some of the photos that we had taken in the lake, and as you can see, we have the water hyacinth and other macrophytes that we have seen. In the Arekere Lake, we found common duckweed, which is mostly used for restoration of nutrients from most wastewater bodies. And then next we found the nut grass, which is cooling in nature and keeps the nearby area cool through its transpiration methods. And from both the lakes, we found one common macrophyte, which was the water hyacinth. Water hyacinth helps in soil enrichment and also provides quality fodder for most of the birds. These are also some of the photos that we have taken from Madiwala.
So we have conducted a survey for all age groups from 10 to 16 plus, and majority of them were given by 25 to 35 year olds. We collected a total of 142
either improvement or decline. Yes. The second question was, what are the most significant threats to the health and the sustainability of the lakes? Pollution and the urban development are known as the primary concerns of the threats to the local lakes. Next slide, please. The third question is how should the local authorities address these sources of pollution to improve the water quality? The better regulation, enforcement of laws, and community education and awareness campaigns are the key strategies which we can develop to improve the water quality. Next slide, please.
So, overall, we can say that the Magnolia lake has a balanced nutrient levels, supports a better quality of bird species and also aquatic life. Then the Arekere lake has higher nutrient levels and also has moderately acidic pH, which is not good for the plants growing surrounding it. It also shows that it has fewer bird species and more invasive plants like the water hyacinth and alligator weed. It also indicates a less balanced environment. And the major threats to local lakes is the pollution and urban development. The solutions to make it better are regulations, public education, and green infrastructure improvement.
According to the 2017 IISC reports, the Arekere lake was more polluted due to the drainage systems and the sewage waste going into the lake. The restoration processes are going on to help to revive the lake. With continued maintenance and efforts, the Arekere lake can be turned into a clean lake such as the Magnolia lake. These are some of the photos. These are some of the photos of the previous years of the lake. And this is the photo after the restoration process has begun. Some recommendations to the solution include enhanced regulation and monitoring, such as implementing stricter regulations to water quality and maintenance, and increasing public awareness by launching new ideas and programs.
See, one of the things you have to do is have two slides. One is why lakes are required and what are the threats to the lake, and also why lakes are required in an urban environment like Bangalore, and what are the threats. Then it gives the continuity and also give there's a who all have worked review of literature, because there are many. Of course, you brought one or two studies of IISC here, but it's better if you can have a detailed thing. Okay, one or two slides on that will help. And the species name, I think you have to be careful, especially macrophytes is incomplete. See, we have put that document on the net which gives the species name, etc. on macrophyte. You have gone to our web page, no? If you go to the technical report section, you have gone through some of them. There's a report on macrophytes itself. And ETR 77 gives you this on how to do the experiment and the lake, water sample, macrophyte, bird monitoring, etc. And also there are videos available, you please refer that. Yeah, since it's See, that is where I said you should go through the literature and then say how people have, what is the values they have got. Then based on that you say which one is relatively better, okay, and which is polluted. Now calling a clean, see, can we call Dwarpalka is a very silent boy? Or that girl has a very, this one, no. We should know their performance in the class and all those things, no. Then we can do that, okay.
Then another thing that is body water, that was microphyte, that is only genus name you are written. So don't put it as a imbalance. Okay. You know the species name scientifically, then it will be
It is a macrophyte, but it is on the terrestrial ecosystem. You are listing the aquatic ecosystem, no? Aquatic means around the where marshy areas and all, that's the thing. The Parthenium and all those the weeds come on the terrestrial ecosystem, okay?
Then you got that question that is based on that is sources of pollution. You didn't mention about the sources of pollution, then after that question you have. The last question that you did the survey, no? In that third bullet that is based on that is sources of pollution, the pollution. Okay, sources of pollution to improve water quality. What are the sources of
No, sources means whether it is by the disposal of the untreated sewage, that kind of thing.
No, then you should, this one you should improve that words. You are thinking about that better regulation and enforcement, that are the things. Yes, yes.
That is the solution that you are using.
Yeah,
So you
solutions.
can apply the solutions.
Solutions for improving the water quality.
You rephrase.
You please read that. Yeah, everything should be very clear. Okay. Because you are the 11th standard now, so you should be very clear.
The question basically meant like how the local authorities should treat these water to improve the water quality.
But you should mention about what are the sources of pollution or such don't know what are the threats. That you should mention in the introduction part. Then it will be good for the this. Why? Because the every slide should be the connection. Yes. That is the important part. Then you will also get the more confident and the presentation also.
So to define a lake like clean and polluted, actually since you are 11th standard kids, so you have gone to Arekere, isn't it? So in there there's a lot of parameters given. So actually as 11th standard student you should be able to check the soil oxygen, like conductivity, there's IISC, chloride, magnesium, calcium, then this alkaline takes up. So if that is, if you do the water quality analysis of both the lakes, then you can compare and tell that it is clean or polluted, which one is polluted and all. So I strongly suggest you have to do water quality analysis of both the lakes. Thank you.
Okay, one more. Tulika.
The images that you have given, what is the source of those images?
We have
Okay. So for that, you have to use the GPS map camera so that we know that you have taken those images. Those are not internet images. Okay. Okay. Next is these maps that you have prepared. Boundary of the lake should be there. Otherwise, how you have calculated the area? Okay. So after you mark the boundary, then you can see how much is the area and the time. Take the validation also. And on the conclusion part, you have cited one IISC report of 2017, right? You should see what is the proper way to cite a particular report or paper. Like that way, you should cite the paper. And one more thing, you have surveyed how many lakes? 2 lakes, right? So in 2 lakes, only 6 birds you have seen.
we have taken the predominant species like I mentioned.
Okay.
We found that if we take predominant species, we can identify it better, like because they, first of all, they are more prone to environments because their presence or absence determines whether the lake is ecologically balanced or imbalanced. So we have only conducted the predominant species, but we will be conducting the more species of birds as well as the water quality analysis as discussed.
See, you use the two words, predominant and also you said prioritize. How did you prioritize based on the, you used two words, one is you said predominant, other one is prioritize the thing. How did you prioritize the thing?
Because if we take a lot of species of birds, then our time duration gets constrained. Right? We are not.
That is different from prioritizing. Prioritizing means if you have 100 students, based on their performance, you say, I selected top 5, that is fine. or the lowest 5. That is also fine. There is some criteria based on that. You cannot say that I selected 5 without any their ecological significance or anything. See, that is where whether they are endemic species or whether they are endangered or whether they are common generalists, that kind of thing they should be. Huh?
And so, like the common birds like crows, kites, that was there, rock pigeons.
Yeah, the common birds like
Common birds were there. So, if you have seen those birds and you are listing the birds, you should also include. That will give you more about the biodiversity, like sir said, na? How you are selecting those. That should be
Okay, very cliche topic of the day, what is carbon sequestration? Nothing but the process of capturing atmospheric carbon dioxide and storing it. Again, it plays a crucial role in limiting climate change because it obviously reduces the amount of carbon dioxide in the atmosphere. Next slide. Right. So there are two major methods in which carbon dioxide and carbon sequestration takes place. One is the biologic method, naturally occurs as a part of the carbon cycle, carried out majorly by the biotic factors of the ecosystem, that is the plants, algae, trees, etc. And then there's the geologic, that is the storage of carbon dioxide in the abiotic factors of the ecosystem, such as depleted oil reservoirs, gas reservoirs, and coal mines that we cannot access, etc. Next slide. Okay, so our study area is our school premises, our school campus, and a lake that is situated right in front of it, that is the Arekere lake. Why did we choose to work here? Because our concern for the environment begins within our immediate surroundings, right? So we chose to work on our school and a lake that is situated right in front of it. Right. So phytoplanktons. Why are we talking about this? Because as I mentioned earlier, our objective is to find the amount of carbon dioxide sequestered by algae, a part of our objective. So algae is nothing but a type of phytoplankton, and planktons are nothing but microscopic organisms, specifically aquatic. And phytoplanktons in specific are autotrophs of the plankton community, that is, they are the producers. Make food, become food is their mode, basically. And quick fun fact, phytoplanktons the word phytoplanktons comes from two Greek words, phyton in Greek, which means plants, and planktos, which means wanderers or visitors. That is basically they float around in water, right? That explains why they got that name. Next. Right. So the phytoplanktons that we observed in Arekere Lake, that is, we collected lake water samples with the help of our security guard from the lake, from Arekere Lake. And we observed the same in our school's biology laboratory. And these were the phytoplanktons that we managed to identify with the help of Google and our teachers, guides, the IISC website, etc. Chlorella, colostrum, chlorobrothum, and microcystis respectively. There's a couple more pictures, but we couldn't identify them, which is why they're not visible. Excellent. Okay, so again, I mentioned for the third time, our objective is to find the amount of carbon dioxide sequestered by algae trees. And these are the methods that we adopted to do so. Dissolved oxygen, that is the Winkler's method for algae, and dehydration using sulfuric acid for trees, about which my friend Amish will be telling you.
Now I'm going to talk about the methodologies that we used to find the carbon that was sequestrated by the plants and the organisms. First, we used the Winkler's method, which is a very common method, to find out the dissolved oxygen that's present in a water sample or any liquid. Next slide. The next slide. After we found the dissolved oxygen in these samples, we used stoichiometric ratios to find out the amount of carbon dioxide that could have been sequestered if this much oxygen had been present in the sample. These are the results of the Winkler's method that we used. From here we can find that the average carbon dioxide that's sequestrated by a sample of 3 liters is approximately 37.24 milligrams per liter. This is the results, right? So here we see that an algal sample can approximately sequestrate 37.24 milligrams. So the sample that we took was 3 liters in water. This is the organism count that we found out in the slides. In the first week, we can see that it is 1.78 into 10 to the power 6 organisms per liter. In the second week, it was almost twice the amount, and in the third week, it was twice the amount. From here, we could see that there was a gradual increase in the amount of organisms in the sample per week. Yes. The second method that we're using is sulfuric acid dehydration method. Sulfuric acid is a very strong dehydrating agent. You can dehydrate almost everything and release pure carbon and other elements that are present in the sample. We use sulfuric acid to dehydrate the twigs that we took from the plant and measure the residue of carbon that we found. From here we found out the carbon content in this tree. From here we can see that a tree which was approximately 147.117 liters could sequestrate about 140 grams of carbon per week. So a tree that is approximately 3 liters, that is the same volume as the container that we took, would be able to sequestrate almost 2,854 milligrams of carbon. This is a comparison slide that shows the comparison between the carbon that's sequestrated by the tree and the algal sample. In the conclusion, we can see that the trees can actually sequestrate more carbon than the algal sample. This is mainly the most probable reason for this is that algae is more primitive than trees. This could be the main reason for algae not being able to sequestrate as much as carbon as the trees. But when we grow algae in large scale such as lakes or containers, we'll find out that it can actually sequestrate more carbon than the trees. Now I'm going to talk about the problems that are faced by the lakes. Lakes are very crucial for human life and many other organisms. Our ancients have been depending on these lakes for many factors such as irrigation and other things. Here we show the problems that are faced by these lakes that has caused major drawbacks in these lakes. So here we see that a lake an average lake that's there in Bangalore has been encroached. So approximately 148 lakes in Bangalore have been encroached out of a survey that we took of almost 160 lakes. Next. So this is an analysis of the encroachment that had happened in the Arekere lake from 2011 to 2024. In 2011, we see that the perimeter of this lake is 1,664 meters, and the area of this lake is 1,17,285 meters square. But in 2024, we see that it's only 1,156.87 meters, and the area is 78,490.31 meters square. I'll explain a few solutions that we can use to conserve these lakes. We can maintain proper septic tanks so that we can pump them in every few years so that the pollution levels in these lakes get reduced. We can also treat polluted water before discharging it into the lakes. We can also plant many native tree varieties and vegetation along these shorelines so that soil erosion gets reduced and the trees and the lake doesn't get polluted. Now I'm passing it over to my friend Dheeraj.
As everyone knows, in big cities there is a lot of pollution, and we have come up with a solution to reduce the pollution. So algae, as my friend told, when cultured in a very large area, can sequestrate a huge amount of carbon, like in par with the trees. And so, if we try to culture the algae in big tanks, which have the enough space that can go for exchange of gases between the atmosphere and the tank, so that the algae can do photosynthesis and so can sequestrate the carbon in this enclosed ecosystem. So advantages of having such setups. So algae is more efficient than trees in carbon sequestration when it is grown at a large level. And also algae, when they are stored in a very when they are stored in tanks, they occupy very less space, and so they can be placed in areas which they don't have so much space, and they have too much of pollution. So it is also viable in areas where planting trees is not possible. So other advantages of culturing algae is that when you have excess algae that is cultured, you can use it for food supplements, and also it can be used as biofuels. So explaining what are biofuels and how they can be used as food supplements. So biofuel is a type of fuel that is grown over a short time from a biomass. So biofuels can be produced from plants or agricultural, domestic, industrial, or the biowaste. So food supplements, microalgae are also a good source of protein, and they are grown both in freshwater and seawater. They are rich in omega-3, omega-7, and omega-6. So summing up the entire advantages of growing algae, so we can see that it can be used as a food supplement. Spirulina, for example, is rich in protein, and chlorella is rich in vitamin B12. Also, we can grow them to reduce the pollution, as they are effective in carbon sequestration. Then we can also use them for consumption, so that we can get a proper amount of protein in our body.
There's a little correction I would like to make. In one of the slides it was mentioned that algae is more efficient in sequestering carbon. That is something that changed in the slide. I'm not sure that it changed. We are not asserting that. That's a wrong statement actually. And now coming back to acknowledgements, we would like to thank our school faculty and our director and principal, Dr. Sinagoy ma'am, our pillars of strength, Gayatri ma'am, Ishwari ma'am, and Divya ma'am, and Navya ma'am more than everyone else, because she's our lab technician, and she put up her comfortably and helped us finish this project, and of course Cincy ma'am and Ganga ma'am for their invaluable guidance. Thank you.
Okay, so we go to second slide. Second slide.
This is the second slide.
No, I wanted that one. For that THO. Then go to the 13th slide. How did you get CO2 sequestered? Can you explain that?
Yes, sir. Sir, we did stoichiometric data. So we tried to find out how much carbon will be sequestered every time.
1 second. You got the biomass, 17.6. Okay.
That is the
concentration of the this one. Okay. Then from there, how did you get the CO2?
Sir, so we use stoichiometry. Stoichiometry is used for measuring the mass of the reactors and products. So we use stoichiometry to see how much, if this much oxygen is getting released, how much carbon dioxide will get released every time a reaction is occurring. So from that we found out
You check the standard method, okay? Then that slide, the hydrogen sulfide, the tree, this one you are talking about, no? What carbon is that you are talking about, inorganic or organic?
Sir, we are talking about the carbon that's getting stored in the tree.
No, when you digest with the hydrogen sulfide, no? Why did you move that? H2S. Just now you had that, no? When you do the digestion with the sulfuric acid, no? What is the carbon you are getting there?
So the carbon is the carbon that's stored as various forms of carbohydrates in the food.
No. You just check that, whether it's organic or inorganic, you have to ascertain that, okay?
Okay.
So, and the other one is you talked about encroachment, no?
Yes.
On the 20th slide. Okay. Where is the encroachment?
Sir, we forgot to mark.
Okay, you forgot. You will do that, no? Okay, then go to 22, the 22nd. See, you talk about septic tank. Are there septic tank in that locality?
Sir, yes, there are septic tanks.
I thought the UGDs are there already. The treatment plant is there. Anyway, check that, okay?
Okay, sir.
Then 23. See, you are using lake for algae. Why you are doing it inside the room, culturing?
No, sir, we are not, we took the algae from the lake, then we are culturing it in a tank.
that means I don't require lake. I should culture it in the this one. Huh?
Despite our efforts of trying to see lakes and the lakes and all those special efforts, this is majorly meant for places, let's say a traffic junction, where planting a tree is not a viable option. In that case,
Okay, fine. Then you said you don't agree with earlier carbon sequestration. See, making the statement, you should have a science-based argument should be. Okay, it is not emotional thing. You should have said, okay, literature shows that. But somewhere you show that as the efficient one, and somewhere else you counter it.
that is what I was saying. Because we had initially told that trees are more efficient in sequestering carbon. In the next slide it said algae is more efficient.
No, that is where the experiment has to be careful. You are measuring the trees, what is the time frame? Algae has a cycling time of every 4 or 6 hours. If you quantify that and include that in the age of the tree, then you will see which one is more efficient. Algae has a shorter turnover, no? The carbon sequestration is much this one, okay? You read the literature. See, one is I suggest you to go through the literature and try to include the review of literature, okay? Relevant to this study, carbon in trees as well as the thing. And what is the method adopted in the standard this one? You should also mention that, okay? What you have done is okay, but still you need to come up with the proper this one. And you should have the clarity on that carbon dioxide this one, okay? Since it's over.
I want to tell one thing that we are not experts. Okay, so we are given here the conference. Then we are not expert. No, whatever in your slide, whatever you wanted to talk, you please talk. Okay, we here only one expert, that is our staff. Okay. So in the first slides, you just going. Don't do like that, okay? Because this is the competition. So, what you wanted to tell, what you wanted to convey to others, that you should do it properly. Then another thing that please take your ID notice. That some clarification in this area. So please take it. That one. Then in your that carbon, that algae thing on you are doing the water samples. Which one you are doing? Cover using the P+ method that provides. That the water sample you are taking or algae samples only you are taking. So your algae or you use the water sample that. That part should be very clearly mentioned. Okay, that is another thing. Then you were then one of the recommendations up here that I saw that algae is more efficient than trees.
that is what I said. That was supposed to be changed. We changed it, I don't know why it's not changed.
Because
Again, she
here
is back
for
to the
carbon sequestration, the algae is also important, the trees also is important. You cannot keep like that sentence in the end of that one. That is, you please remove that because in your based on your sentence, we want you to cut off all the trees and that it is not like that. But your that is words, that sentence is given like that. Okay, so don't
So there are very good literatures are there. Go through that. Okay. Okay, Aparna.
So first of all, in the introduction part, it would be better if you can make a carbon cycle of lake. Like here you mentioned about, I think it's a terrestrial carbon cycle. You can make a carbon cycle of the lake. You can take the lake picture and incorporate what are the factors which will contribute to carbon sequestration, like the macrophytes and the microalgae. Microalgae is phytoplankton. So then the water body, then the sediment. So it will be great. So you guys will get an idea regarding the carbon cycle. Then after the carbon cycle only the carbon sequestration, the topic will come. Okay. After that, I felt in that introduction part you
One
can.
second. Before that, which is that animal?
That is like fossils are there.
Huh?
So yeah, and you can also mention the photosynthesis equation, no? Please incorporate that the photosynthesis equation. So you get to know regarding the biomass, what like aspect like organic and inorganic. So the organic matter which is incorporating into the body of the like plant or animal, the organic form it is there now. Like sir asked there like few minutes ago what carbon is sequestering. It is the organic carbon. So you should be thoroughly know these things. Then coming to the slide 8, you guys please mention the essay unit. The slide 18, I guess. Yeah, 18. Mention the essay unit and also the different experiments you have done now, like sulfuric acid dehydration. Please cite like which literature you have referred. Please cite the literature. And then like collection, regarding the collection of algae, how you guys collected, and enumeration, also the enumeration part, like Sedgwick Rafter, which method you guys did, how you collected the algae samples.
Did you collect algae?
Sir, we took the lake water. From the lake water, we started culturing the
You guys
started
pulse.
adding the fertilizers and the salts that are required for algae growth. So we started gradually. But it was before.
Read the literature. Okay.
Like
follow the standard protocol.
why? Because if you are culturing it in another media, which means it is not like
It's not
proper.
lake.
Yeah, it is not like no. And how you cultured it? I'm just curious. That's it. Because culturing which need like inoculum from this thing, from the lake sample, right? I thought that images from that liquid trees, what your culture that.
That is not what we actually. We collected water samples. Then we just
It's okay.
yeah. So we got the water samples and we kept it in our biolab. We observed it for I think 3 weeks.
Okay,
We didn't give any source.
Okay.
No transformation. We didn't change the water. As it is, we just kept it and observed it.
Okay, at least, okay, so the water level, it is not reduced, it is 3 weeks, the water level. Okay. So, like you guys have to go through the, like the methodology part. There are many literature, how like they have mentioned how they are collected, how they are cultured. If you want to culture, otherwise you can directly take the algae sample from the lake itself and do the test, okay? And also go through the experiments, go through proper like APH is there, like then Waki Black experiment is there. So go through all those kind of
Let
experiments.
them go through the thing. You wind up now.
That's it.
Thank you.
Okay, here there are students and teacher corners are there. Okay, you can refer that. See, in this, before there's an orientation program, what we did, all those details are available here. You know, the presentations, what we did, school wise we have put it so that you can go through that. Okay, suppose if you take this, BGS you had come, no? Each one of us, our presentation videos are available. You can have a look at it. Then after this one, the evaluation, we are going to put what you have presented in this section. Okay, similar to what we have done for the other school. Okay, that is not coming. Delay. Okay, see, we have done in Arekere, that presentations we have put. These are all the presentation what children have done. Okay, their slides as well as the one. Okay, this is as far as the conference is one. And this is the reference material which I said. See, here there is a environmental monitoring. The children who are working on water quality etc. can refer that. Okay, and there are other informations are also there. Information on plants, butterflies, birds etc. You can refer this section. Okay, and if you want to see the other details, you can go to our website. There is the research group. Our research papers are also available here. The research paper sections here and here will give you the theme wise research paper. See, these are all related to the lakes. You can refer that. Okay. Now with this, I request Tulika to talk about the maps. Okay.